Hello, everyone, and welcome to today's webinar on overcoming challenges in antibody discovery. I'm Alyssa, and I'll be your moderator for this session. We're delighted to have you with us for what promises to be a very informative and engaging event. It's my pleasure to introduce our speaker for today's webinar, Dr. Jessica Hartman. Dr. Hartman is the Senior Director of Applications and Lab Services at Cell Microsystems. She brings a rich background in biology and molecular cancer biology, holding a Bachelor of Science in Biology from the University of Virginia and a PhD from Duke University. Her postdoctoral training spans biochemistry and cancer biology at both Baylor College of Medicine and Duke University. And with extensive experience in directing bioscience research and development for biotechnology companies, Dr. Hartman is now leading the charge in developing new and streamlined workflows at Cell Microsystems. Before we start, a few quick points. We encourage you to submit your questions at any time using the Q&A feature, and we'll address as many as we can during the live Q&A session. In case we cannot get to all of the questions, we'll respond to your inquiry via email. Also, this session is being recorded, and we will be sharing a link to it in a couple of days. With that, I'm excited to hand over the virtual stage to Dr. Hartman.
For that kind introduction, and I cannot thank all of you in the audience enough for joining us today. I'm so happy to have you here to introduce you to our brand new application using our technology to accelerate antibody discovery and screening. This has been several years in the making, and I'm very excited to debut it today and launch it into the scientific community. So during today's webinar, I'm hoping that these are the learning objectives that you'll take away from our time together. We're going to spend a brief time introducing the role of antibodies in research and medicine. I'll talk about some methods for antibody discovery and screening, as well as some advantages and disadvantages of each of them. And then I'll introduce you to our novel approach for upstream sorting and functionally characterizing antibody-producing cells using our technology. I'm sure if you're joining us today, you are very familiar with the role of antibodies in discovery and research, but I thought it would be good to get us all on the same page about some of the definitions. Outside of the immune system, antibodies have taken hold as very useful tools in both the research and the therapeutic space. The class of antibodies that are typically used in research are used for targeting molecules to study basic functions of cells in vitro in a dish, or to create diagnostic or testing assays, for example, such as the COVID-19 test that we're all very familiar with. Although research antibodies are very useful just in the lab for basic science, they can also be important for informing the other class of antibodies, which are the therapeutic antibodies. Therapeutic antibodies are used in the clinic to treat patients. They are targeting molecules that are critical for the progression or the onset of disease, and these therapeutic antibodies can be used to either treat or ameliorate or prevent the onset of disease. I like to show this chart here because I think it really emphasizes the explosion of the role in monoclonal antibodies in the therapeutic research space since their development in the mid-1970s. On the left-hand side of the graph, you can see the first monoclonal antibody was launched in 1975 and had begun to be entered into the clinic for the use of treatment of a variety of diseases. There's been an absolutely meteoric rise in these antibodies in the clinical landscape ever since, with the market value of these drugs reaching into the billions in the modern era. Monoclonal antibodies have been used for a variety of treatments, ranging from oncology to asthma to osteoporosis. But what I really want to draw your attention to in this graph is what I would consider to be the inflection point in the use of monoclonal antibodies in the clinic. And that's the advent of trastuzumab or Herceptin in 1998. This was the first targeted antibody for use in breast cancer patients that were expressing the HER2 gene. And while I could stay here all day and talk to you about the dollars and cents of antibodies and their use in both research and therapeutics, there's a very real human factor to the role of these antibodies in the clinic. Herceptin was hailed as the drug that changed the breast cancer treatment landscape. Since 1998, when it came out, it has saved over 3 million women's lives. And one of those women is pictured here right on this screen. This project is incredibly close to my own heart because that woman is in fact my mother-in-law. She was diagnosed with HER2 positive disease over 5 years ago. And women who received a diagnosis of HER2-positive breast cancer had initially one of the worst prognoses when faced with this diagnosis. Now, women like my mother-in-law who have HER2-positive disease have a greater than 95% 5-year survival rate. So you can see that these aren't just antibodies in a dish, they have a real human impact on the ability to make people's lives better and to create real change in our therapeutic landscape. Despite all of the benefits of these antibodies and all of the advances and the progression in the clinic, there remains a very long process in the journey from the bench to the bedside. As you can see in this commonly shown type of workflow, going from research and development to review and approval and actual use of a drug in the clinic can typically take upwards of a decade. Although there are a variety of bottlenecks along this pipeline, one of the biggest hurdles is just the beginning of this pipeline in the research and development phase. Identifying antibodies, screening them, doing lead identification can take years in order to find candidates that have any chance of being efficacious in the clinic. So what I'm going to focus on today is how we shorten this early phase of development to identify the best potential candidates early in the process and fill that pipeline with actual targets that have a real chance of progressing into the clinic. So when you're starting to look at how we go through antibody discovery and expression, it's a very long process. And you're going to see this funnel throughout the talk because I think it's a really important anchor for understanding antibodies in both research and disease. When you're trying to find a target antibody, you have to go through multiple rounds of discovery where you're looking for expression and specificity. You then have to characterize those antibodies, optimize them, and then do candidate selection prior to going into preclinical testing. So it's very important at the top of this funnel that it's filled with lots and lots of candidates that can survive the attrition process that it takes to get through this very long pipeline. The fuller the funnel and the better the hits, the more likely it's going to be that you'll have an actual candidate at the end of this process. So how do we fill an antibody funnel? There are several different methods of antibody discovery that are currently utilized, and they have advantages and disadvantages depending on your needs. Perhaps the best recognized and the most common of them is hybridoma screening. It has the advantage of producing high affinity antibodies. You get paired heavy and light chain antibodies right out of the gate, and it's a fairly straightforward and low cost way to identify antibodies for your downstream workflows. The disadvantages of hybridoma screening is that due to the way that they're made, these lines tend to be genetically unstable. You typically have a limited antibody diversity due to the way that they're produced, and it's a very labor-intensive process. It is, however, the gold standard in the workflow, and it was a long-standing staple of antibody discovery until phage display came into the arena. Phage display has the advantage of being high throughput. It yields the genetic sequence of the antibody, and it can be used to produce fully humanized antibodies out of the gate. The disadvantages of phage display are that they're typically size-restricted libraries, again, meaning that your diversity is fairly limited. And unfortunately, due to the fact that you're only expressing a fragment of the protein, you don't get the post-translational modifications that you would see in the hybridoma screening workflow. More recently, in the advent of more high-throughput sequencing technologies such as Sanger sequencing and next-gen-seq, we can now perform B-cell screening directly on the antibody-secreting cells. The advantages to this method are that we can interrogate a diverse pool of targets from a large sample size. You do retrieve the direct antibody sequence, and it's species-specific to whatever B-cell input you're putting into your workflow, whether it's animal or human. The downside to the B-cell screening workflow is that B-cells are non-proliferative. They do not survive particularly well in culture, and so you're limited by which cells survive the processes that you're subjecting them to. There is no functional readout from this assay until you're very far down the pipeline, meaning you're potentially looking at thousands of cells that are not target specific, and it can be very expensive depending on the scale that you're working at. Now, despite having very different workflows, the one main feature that all of these methods have in common is that no matter where you get your antibody or your sequence from, you then have to embark on the antibody screening cycle. After you have found cells that are antibody producing or expressing or have been engineered to express your antibody, you now have to determine whether or not those cells are producing functional and specific antigen targeted antibodies. To do so, you're typically taking a cell line and you are limiting diluting or pipetting or robotically pipetting cells into microwell plates like this. You're then performing ELISAs and looking for hits like you see in the cartoon in a plate reader type assay, and then doing informatics to distinguish your specific binders from your non-specific binders. This process is typically a rinse and repeat, requiring several rounds of enrichment in order to find monoclonal antibodies and to determine specificity. And if you've ever done a plate-based ELISA, like I'm sure most of us have, this is not an insignificant resource, labor, and cost burden, taking many plates, many tips, lots of media, and screening things that may not even be accurate. So overall, this antibody screening cycle, no matter how many targets you have, ends up being resource, labor, and cost extensive. It's very inefficient. And worst of all, you're almost always screening blind, meaning you're probably screening thousands of cells that are not even the cells that you want. So I'm going to use this stack of 10 plates throughout the talk to kind of anchor us to how I think our technology differentiates us from the typical workflow, where you're using these 96-well multi-well plates for your screening. If you're lucky in one of these assays and you're only handling a plate of 10 with single cells in each well, you might have upwards of around 960 cells in a stack of 10. That's assuming every single cell survives and you have the ability to screen all of those cells. So in 10 plates, you're screening less than 1,000 cells in over 100 mils of media. So in a world where we're trying to fill that funnel as full as we can get it to maximize our chances, 1,000 cells doesn't feel like enough. So this is where our technology comes in. Our consumable, the cell raft array, is shown in cartoon over here on the right-hand side. In contrast to a multi-well plate, has a very small form factor. It's the size of a 60-millimeter cassette. And within that consumable, we can seed thousands of cells within individual microwells to interrogate cells on a single cell basis. Within one of our consumables, we can screen upwards of 40,000 cells per array in only 3 mils of media. To compare apples to apples and make it fair, in one of our consumables, if we can screen 40,000 cells, that translates to roughly 496 well plates and over 4,000 mils of media. So I think I'm going to show to you, what I'm hoping I'm going to show to you as we move through these case studies is the advantages of screening more cells upstream in a small format for downstream analysis. We do this using our automated and efficient antibody discovery pipeline, which combines the consumable, which is the cell raft array, that allows us to do upfront antibody characterization and single cell separation, with our software, cell raft cytometry, and our instrument, the cell raft air system, which is how we actually perform the image-based target cells of choice. You're going to see several cartoons like this as we move through the case studies I'm going to show you today, but I thought it was important to anchor you on what we call in-raft antibody production screening. It is essentially a miniaturized ELISA or antibody binding assay within each of these individualized microwells. The assay is incredibly flexible and very easy to execute simply by combining different fluorescent detection reagents and conjugated antibodies. What you can see in these cartoons is 3 potential readouts in the assay. We can add into the media of the microwells antibodies that can capture generic IgG, as well as assess for specific antibody production using biotinylated antigens and conjugated streptavidin secondaries that have a fluorochrome attached. So in this workflow, if you had an antibody-secreting cell that did not have any antibody at all, you would see no fluorescence signal like in the right-hand side. If we had an antibody-secreting cell that had a non-target IgG or we weren't looking for a specific target, you could see 1 fluorochrome. Or if we were looking for target-specific expression, you would see a dual fluorochrome expression. And what that will look like in sort of a cartoon fashion is essentially a heat map on each microwell position of on a single cell level or on a clonal level, what antibody is being produced in each of those microwells. So for the remainder of the talk, I'm going to show you how we have leveraged the assay that we've built in 3 common antibody discovery workflows: hybridoma screening, B-cell screening, and then ultimately recombinant antibody production screening. So the first case study I'm going to talk to you about was using hybridoma cell lines. To give you a little bit more information on how these are actually done, they start by immunizing an animal of choice. In this case, a mouse that can be immunized with an antigen to stimulate antibody production. After the mouse has been allowed to stimulate the antibodies, you can isolate the antibody-secreting plasma cells, and then they undergo a fairly impressive fusion process with a cancer cell line called a myeloma. This step is absolutely critical because plasma cells, as I've mentioned, are non-proliferative, meaning that despite the fact that they're antibody producing, there's no way to upscale or interrogate those antibodies because the cells are not going to survive. So when this method was developed, they leveraged the power of the cancer cell to be infinitely self-replicating with the B cell production of the plasma cell to create these fusions called hybridomas. And in that way, you get the best of both worlds. Antibody production, but long life and a long span of antibodies being produced in your supernatant. Through a very elegant selection process in specialized media, you can screen for the cells that have undergone that fusion process and against any cells that have not. And then the cells are single cell arrayed in a plate like this and assessed for functional antibody production. Once you've identified hybridomas of choice, they have to be expanded up to produce your monoclonal antibodies. As I mentioned previously, this process is fairly labor intensive and has a pretty low efficiency. But even if you get to the point of expanding a selected hybridoma that is producing your antibody of choice, as anybody who's ever cultured cells in a dish knows, over time with operator changes, cryopreservation, upscaling, these cells can genetically drift from the parental cell line. So even if you went through the effort of making a clone, over time, it's entirely feasible that you'll eventually lose that antibody production phenotype. And that is exactly what happened in the case study I'm going to show you now. Tetracore is an antibody production company in Maryland. They are a good collaborator of ours that reached out to us for help with this exact problem. They develop kits, assays, reagents, and instruments for the detection of diseases, biological warfare, or biological threats in the field. And their kits are intended to be used in the field with first responders to assess threats like this. They reached out to us because one of their best-selling kits, to botulinum toxin A, had actually been pulled from the market due to the fact that their hybridoma cells had a decreasing antibody yield. You can see that in the graph from Tetracore below. In the blue line along the x-axis, you can see that the QC pass lot has a very high absorbance and a high antibody titer. In contrast, one of their recent lots failed to pass quality control because the cells were no longer producing antibody. So they reached out to us as a collaborator to test out our workflow to see if we would be able to rescue the hybridoma phenotype and recover their antibody producing cells. So what we did in our lab was to assess the BOT-A hybridoma cells for antibody secretion. We seeded 20,000 of the parental hybridoma cells on a single cell raft array. This looks like you see in the cartoon here, with the BOT-A cells represented by the orange IgG secreting cell. And to assess whether or not they were actually making antibody, we simply spiked in a FTSE conjugated anti-IgG secondary. In the event that the cells are actually producing IgG, you'll see a green fluorochrome signal like this. The assay is very straightforward. All you have to do is add the conjugated antibody directly into the media and look for green fluorescence, making it very straightforward. No washing steps, no centrifugation, just basically mix and add. When we have the cells ready to go, they are seated on the array in a minimal volume of media. We then scan them to look for single cells at time point 0, and then assess them for antibody production over the course of about 24 to 48 hours, depending on the line. And then we use our instrumentation to recover those positive clones for expansion and assessment of IgG-specific secretion. So what you can see here in this field of view is a bright field image of just one representative field of view of the cell raft array. And what you can see as evidenced by that red arrow in the middle is an example of a cell raft containing a single cell. As you've probably noticed, if you're looking around, some of the rafts contain single cells, some might contain doubles, but the beauty of the software is that we can see all of those cells and we can watch what happens to them. When we perform a second scan, you can see that those cells continue to grow and proliferate, meaning that they're viable. And then we can actually do an assessment of the FTSE staining. And so I think if you were paying attention, you would notice that despite the fact that all of those cells in the bright field, there's only one lone cell raft here showing any FTSE expression. And the first time we saw this, it was absolutely mind-blowing because it really illustrated how rare their antibody cells had become in that parental population. So each of those green speckles represents the fact that the produced antibody is being bound to the detection reagent and in fact is localized to that raft of choice. To zoom in so you can see this a little bit closer, we can see the single cell at day 0 with that antibody positive signal at day 1. And those cells can then be isolated and expanded for downstream testing. So we're not just looking for antibody production. This raft with the cells on them is fully recoverable, and the cells can be grown off of the consumable for downstream ELISA. Now, that one lone raft sitting in that field of view is great, but as most of you are probably thinking to yourselves, that's one raft in a sea. How on earth do I find that raft in order to recover it and expand it? And this is where the software comes in. Because all of our data are image-based data, we can use our software to very easily identify rafts containing our cells of interest. So we can use the software to identify positions containing a single cell at day 0. We can look for FITC expression, as you can see here at day 1 on the right-hand side. And then the software can take both of those population sets, overlay them in this lovely Venn diagram that you see in the middle. And the intersect, highlighted in green here, represents our targets that are single cell clones that are in fact antibody producing as measured by green fluorescence intensity. Overall, the software was over 98% precise at finding those needle in a haystack rafts containing the clones of interest, and those clones could then be recovered and transferred for downstream expansion. You can see what that expansion looks like here. If you pay close attention, you can see the square in some of these. That represents the raft that the cells were attached to. And those hybridoma cells have gone right back into cell suspension after they continue to grow once they've been transferred. This image is only 7 days after the transfer of the raft to a 96-well collection plate. We were able to expand 13 clones that were antibody expressing, expand them, and then screen them downstream by ELISA. And I quite like this picture because you can see here that we have 3 different clones from 3 different wells, and they each have radically different growth rates, even though they all started from a single cell. And I think this really highlights what can happen to cells over time as they begin to drift and their phenotype begins to change, even in a population that was once clonal. So having this level of screening power allows us to not just settle for cells that are growing, we can actually find the cells that we want that meet our best characteristics and continue to use those. We were able to use TetraCore's actual commercially viable kit to test the antibodies that these clones were secreting, and that's the data that you see here on the graph. Each of the clones is represented on the x-axis, and the fold increase above background is relative on the y, with the parental shown here in 0. So as the parental cells were expressing absolutely no antibody, they gave no signal in TetraCore's commercial antibody kit. In contrast, all 13 of the clones that we isolated gave greater than 10-fold expression of the antibody in the assay that we used to assess function. We then transferred those 13 clones back to TetraCore for assessment in their own lab and ultimately to purify the antibodies and to requalify them. And what you can see here in this graph is just the supernatant from those clones tested by TetraCore relative to their purified commercial antibody. And I want to draw your attention to that little asterisk at the bottom. Each of the supernatants from the Cell Microsystems clones had to be diluted 1 to 1,000 to make it onto the scale relative to purified commercial antibody. So you can imagine exactly how much antibody is being pumped out by these high producing clones that we were able to rescue. In addition, you can see that each of these clones are very close together in terms of their antibody production, suggesting that the cells that we're recovering were once upon a time clonal and that we've been able to retrieve them from this drifted population. Lastly, Jen at TetraCore purified the antibody from the clones that we provided, and take you back to the graph that I showed you at the beginning. That blue line representing their last passing commercial lot with the gray line representing the failed commercial lot. And we were just absolutely over the moon to see that the cell raft purified antibodies from the clones that we provided to TetraCore had a comparable or better performance than their current commercial lot. I think this quote from Jen says it all. As she says, the purified antibody shows remarkable improvement over the previously purified lots pre-cell raft. They've actually tossed out all of their old clones, and they gave these new clones a cell microsystem ID, which just tickled us pink. So we were really grateful for the opportunity to work with TetraCore and to help them get their kit back on the market and to test our workflow with a legitimate commercial customer. So to really highlight the scope of what we did in this workflow and the importance of filling that funnel I told you about at the beginning, in this workflow, we started with 20,000 parental cells. On our consumable, there were only 68 positive events for antibody secretion. That represents 0.34% of the total population that was still antibody secreting. Of those 68 events, only 27 of them were clones, which means of that 20,000, only 0.1% was clonal. Even after isolation, only 13 of them still had proliferative capacity and were able to expand and grow and produce antibodies. So that means all combined, the attrition through this funnel is 0.065%. So you can imagine how Herculean this would be with technologies screening fewer cells to find those rare producers that are still existing in the population. This looks pretty bleak. These numbers in red are a little scary, and it does suggest that the workflow, still has attrition. But what I want to draw your attention to is how successful this was. Of those 20,000 cells and those 68 events, 40% of them were in fact clonal. Of the clones that we isolated, 48% of them were able to expand. But this is the number I'm most proud of. 100% of the expanded clones were in fact antibody producing greater than 10-fold. Not only were we able to salvage and recover these cells, but we had 100% accuracy rate with our assay's ability to detect those antibody secreting cells upstream of the workflow before all of the effort had gone into characterizing the cells downstream. All told, it's a very efficient workflow. From thaw to characterized cell banking took only 28 days, and the hands-on scientist time was just over 21 hours. So to put this in terms of that stack of plates I showed you at the beginning, in a single cell raft array, we were able to identify 13 antibody-expressing clones, which is only 2 columns of 196 well plate. If we were going to use the numbers I just showed you to extrapolate to how many wells of 96 well plates we would need to recover, this translates into 16 96 well plates to get only 1 clone. And not only is that a really low number, you still have no guarantee that every cell that you put into these 96 well plates is going to survive. Not to mention the labor and time investment that is involved in assessing every single one of those single cells for downstream function, rather than assessing only the 13 hits that we found on the air system. So after the success of this hybridoma workflow, we wanted to turn our attention to single B cell screening. All of this work was done in collaboration with our co-PIs at Duke, who allowed us to use their animals and their workflow to validate single B cell screening on the platform. If you're not familiar with single B cell screening, it starts the same exact way that the hybridoma workflow does. You immunize your animal of choice with antigens to stimulate production. You then isolate the antibody-secreting plasma cells, but rather than doing the fusion process in a hybridoma, you then have to single cell segregate using a sorter or limiting dilution in order to enrich for those B cells. In this workflow, each individual well gets a single cell. At that point, you then have to undergo RNA purification, reverse transcription, and then PCR and sequencing. And as anybody who's ever done this on a small scale knows, even that process alone, you're going to lose information. Not every cell is going to be recoverable, not every cell will survive, and you may not even get genetic information from all of those cells. In addition, at this point, we still have very little information whether or not any of these B cells are actually specific to what we're looking for. So then what you have to do is take those sequences and engineer them into expression cassettes that can then be transfected into your cells. Once cells express the transient transfection, they can then be screened using ELISA or plate-based methods for antibody production, just like we saw before. But really here, the bottleneck is that you are screening thousands and thousands of sequences at a very high cost for potentially very limited information or absolutely no target specificity, which you would not know until you got all the way down to step 6. So what we wanted to see is if we could flip this paradigm on its head. Rather than starting from B cell sequencing and trying to go backwards to antibody secretion, we wanted to see if we could identify antibody secreting B cells prior to sequence recovery. We can do this using the array, because we don't need the cells to grow or proliferate. We can interrogate them quickly in situ in the array for antibody secretion and then isolate them for downstream analysis. This workflow is a little bit more elaborate because in this, we're actually looking for antigen-specific antibodies. At Duke, they immunized the mice against a protein called ovalbumin, or OVA, as I'll refer to it. We chose OVA because they were really just trying to validate the workflow. It's a readily available protein. They use it in their own lab, and they were looking to make an antibody source for OVA. So the mice were immunized against ovalbumin. We isolated spleens from the animals, purified the plasma cells through magnetic enrichment, and then seeded the plasma cells on the array with detection reagents. In this version of the workflow, we add in the conjugated anti-IgG FITC to find non-target IgG. We also add in antigen capture beads. This allows us to capture proteins to help us enrich a signal which might potentially be low from just a single B cell in the microwell. And then we also add in biotinylated OVA, which can bind to conjugated streptavidin, which has a red fluorochrome. So in this assay, as I showed you earlier, red would mean OVA antibodies, green would mean generic IgG. The workflow is nearly identical to the one that I just showed you. Seed single cells on the array in the presence of these different reagents, scan for single cells, interrogate for fluorescence on the air system, identify the clones of interest, and then transfer them. Now, the main difference here is that we don't really need the B cells to proliferate like we need the hybridoma cells to. In this workflow, we use the air system to isolate those positive rafts directly into 96-well PCR plates into lysis buffer, at which point the plates can be processed for nucleic acid and sent for downstream sequencing with whatever modality you use for analysis. So as you can see in this bright field image on the left-hand side, another field of view of what it looks like with the B cell screening. All of those really dark spots that you see here represent the beads that are present in roughly every single microwell to help us capture that antibody-specific signal. If we look at FITC expression in this field of view, you can see several rafts in the FOV that are FITC positive, suggesting that the B cells in this field of view are actually capable of secreting at least some degree of IgG. At this juncture, we don't know whether or not it's targeted to OVA. When we add in the red fluorochrome, it's very easy to see that one beautiful raft down there at the bottom. And suffice it to say, it was a red letter day for us when we saw that red signal as well. Using this fluorescent assay, we're able to determine that the B cell in this raft is secreting OVA-specific IgG, which of course gives us a signal in both colors. To show you what this looks like more zoomed in on a raft by raft level basis, you can see a non-target cell here in the cell raft in the presence of the beads. And you can see the beautiful green fluorescence suggesting that the antibody is being secreted by that B cell in the top and it's being grabbed onto by those capture beads. We do not see any red signal in this raft because it's not an OVA-specific cell. In contrast, I think it's quite clear that these B cells shown here in these other three examples are secreting OVA-specific IgG, as evidenced by the presence of that red signal being captured onto the beads. And interestingly, even within these three different B cells, we can see varying degrees of antibody production, even amongst the three different specificities. So in order to confirm that we were actually finding B cells that were specific to OVA and that we were not just finding red non-specific binding, we isolated those OVA-specific and non-specific B cells. We sent them off to a third-party vendor for sequencing. They recovered the IgG heavy and light chain sequence. Due to cost and timing purposes, we decided to only send 7. We sent 3 non-targeting IgGs that were non-OVA specific and 4 OVA specific B cells that that had shown red fluorescence on the array. The vendor that we worked with was able to identify those IgG sequences. They cloned them into an IgG2A expression vector, and those vectors were then transiently expressed in CHO cells for recombinant antibody production. They purified the antibody and sent it back to us for downstream analysis. And if you look at this heat map on the right-hand side, you can see what the spectra of sequences of those antibodies looks like. The green boxes above are the non-target IgG, and the red ones are the 4 OVA-specific clones. What's quite interesting is that 2 of the clones that we recovered and sequenced actually have identical heavy and light chains, as evidenced by those purple boxes. In contrast, the 2 on the bottom have identical light chains, but have different heavy chains. So because of the differences in the sequence, we decided to go ahead and synthesize antibody from all of these. And what we were absolutely gobsmacked to find is that we were able to successfully identify ova-specific secreting B cells and to make recombinant ova-specific antibody with the sequences that we recovered. In that green box on the right-hand side are clones B7 and E7. They are the ones that had the identical antibody sequence. You can see that the antibody that was produced from the vendor has identical reactivity to the commercially available antibody that we tested it in combination with. So that blue line on the top is a commercially available OVA antibody. The yellow and the green lines are the antibodies that were synthesized from our B cells. Now, what was interesting is that those other two OVA-specific clones that had the same light chain but different heavy chains, neither of them showed any reactivity in the downstream ELISA screen that was performed by our collaborator. There's a lot of different reasons that this could happen. Obviously, producing antibodies in a producer cell line is not straightforward, and it's entirely conceivable that something about those sequences did not properly express or produce antibodies. But I do think it's really fascinating that the two antibodies with the identical sequence both performed extraordinarily well with the exact same antibody expression compared to the other two. And again, I think what this underscores more than anything else is the importance of having a large number of possible candidates in your funnel so that you can deal with the attrition that happens as you begin to winnow down to your antibody of choice. So I think this funnel we call finding the needle in the haystack, because when you think about the scale that we had to go through to interrogate these B cells to find a single antibody sequence, it's fairly staggering. We started from several different mice, in this case 3 mice. From those 3 mice, we were able to recover 285 million splenocytes. Through bead-based enrichment, we recovered 52 million B cells, which is 18% of the total primary population. Of those primary B cells, further enrichment for the plasma cells yielded 249,000 plasma cells, which is 4.7% of the B cells. And ultimately, from those 249,000 cells, all of which were analyzed in the assay, we were able to recover only 36 putative OVA antibody expressing B cells, which represents 0.014% of that 250,000 cells. So it's an incredibly small number, which ultimately turned into 4 IgGs that we interrogated, one of which validated, which means that only 0.001% of the plasma cells that we analyzed were validated for IgG expression. Now, of course, we did not assess all 36 of these. We certainly could have. But the important point here is that we were able to enrich this very big population for a very small number and recover sequences that were actually validatable. We were able to recover those sequences and test them with our collaborator. And from plasma cell isolation from the mouse to nucleic acid generation and being sent off to the vendor for synthesis takes 2 days of hands-on time in the lab using only 5 hours of scientist time. And of course, this can be scaled as you wish to screen as many plasma cells as you have access to. So to go back to what the arrays and the plates look like, in this workflow, to screen 250,000 cells, we needed 4 arrays. In those 4 arrays, we recovered 36 clones, which went into less than 1 96-well PCR plate for sequencing and cloning. In contrast, to find a comparable percentage, to find just 1 clone, you would need 75 plates containing single B cells. This is almost unbelievable to me that it would even be conceivable without lots and lots of robots and lots of effort. And I think the thing that's most important in this B cell workflow is that you would spend a lot of money and time getting sequences for things that would largely be non-specific. Whereas in the cell-raft air workflow, we can upstream enrich for antibody secretion long before you ever have to sequence anything. So what do you do once you have that sequence? Well, the next step, as you're probably aware, is to make antibodies for recombinant antibody production. In both the research space and the therapeutic space, you have to mass produce these antibodies to have any real commercial or scientific value. So the sequences can then be cloned into heavy chain expressing and light chain expressing vectors. Those get transiently or constitutively expressed in your producer cell line of choice, typically a CHO-K1 or a CHO-S or a 293T. You then have to stably screen for integration of your antibodies, or you have to screen for a marker if they have a marker, and identify which of the cells are putatively expressing your antibody. At this point, you're making cell stocks, potentially of things that may not even have antibody secretion, and this typically repeats for multiple rounds until you've created a monoclonal colony that you can enrich for. Once you've identified cells that are secreting your antibody, they have to be scaled up, the protein has to be purified, and then the antibodies undergo multiple rounds of ELISA-based control to determine whether or not you actually have functional and specific antibody. So we wanted to see if we could use our workflow to help us identify these high producers on the stable cell lines that we would then result in once you found a sequence of choice. So to do so, we worked with our collaborators at Duke again. They were trying to upscale production of an antibody for the mouse version of Herceptin. They took HEC293 T cells and infected them with a lentiviral construct to overexpress their antibody. In this particular version of the workflow, the cells are already GFP positive. And so we switched that conjugated fluorochrome to be a red fluorescent antibody. So in this case, green does not indicate IgG, green indicates the cells, and red indicates the expression of the specific IgG. And now what's interesting about this project is that they had already done some work to enrich the population for GFP. So rather than just relying on the bright field or relying on ELISAs, they had done some upstream effort to get a presumably pure GFP population by flow sorting. And they then interrogated them for antibody production. In this assay, they seeded their GFP enriched cells on the array in the presence of the detection agent. Just like before, cells were seeded, the reagent was spiked in, and the cells were assessed for RFP and GFP on the air system. And then over the course of several days, they were monitored for clonal propagation and then isolated for downstream expansion and characterization by ELISAs. So what you can see in this FOV is now no longer finding a needle in a haystack, it's finding the hay in the haystack. We're now looking at a cell line that has been transduced with a vector of choice. And there are lots of clones throughout the array, all of which are GFP positive. But as you can see on the right-hand side by the antibody expression, despite the fact that there's lots of GFP, not every cell on the right-hand side, when you look at the red antibody secretion, is expressing equal or even any antibody at all. So we were curious whether or not we could use the software to identify our highest producers. And so what you can see in these two images that I'm showing you first are two different examples of single cells on day one. And I think we can all agree that the cell on the top has demonstrably less GFP expression compared to the cell on the bottom. So what I like to highlight here is that even after flow enrichment, GFP positivity was not necessarily all on one end of the spectrum. There is still a difference in the population in the level of expression that you get even after flow enrichment. And unsurprisingly, that cell that was expressing very little GFP expression, despite the fact that it continued to grow and maintain GFP, it also had very little antibody secretion as measured by red fluorescence. In contrast, that bright green cell did expand quite nicely, maintained its high level of GFP expression, and did secrete marked antibody as measured by those red punctate spots that you see here in the middle of your screen. Now the plot thickens a little bit when we start to look at what GFP high fluorescence actually means. Here's another example of a clone. You can see in this particular instance, you might argue that the cell on the bottom has more GFP than the cell on the top. But we're beginning to reach the limits of detection of the computer, flow cytometry software, and the human eye. It would be very, very difficult to use the GFP fluorescence to start to say which of these is brighter or less bright in any common segregation method. When we look at the antibody secretion of these two cells, even among bright green cells, we can see a very different expression of the antibody that's being secreted relative to these two clones. Again, underscoring the need for using these types of assays up front before beginning to go through all of the effort of expanding a clone that is not potentially producing your highest level of antibodies relative to other cells in the population. This becomes even clearer if we look at the quantitation of this phenomenon. We can use our software to actually count how many cells fall into each of these buckets. As you might expect, cells that express low GFP almost never have any high expression of antibody production. Those GFP low cells can have low antibody production. So you could draw a line very easily between the rows in this table and say, I can distinguish between my low and my high, recognizing that my high cells will have more antibody. However, the RFP high cells that are producing the highest amount of antibody in this bucket are completely indistinguishable from the RFP low relative to the GFP high cells. So within the software, these GFP high cells look exactly the same regardless of RFP high or low. And so what this tells us is that it's important to not rely on any single marker and to actually use this upstream screening in order to make our downstream expansion worthwhile. So at this point, we use this funnel to kind of emphasize the fact that we're not just finding the hay in the haystack, we're finding the best hay in the haystack. Even in a pre-enriched population of GFP sorted cells, there was a wide variety of differences in the cells that we interrogated on the array. Of those 50,000 293 T cells that went into our funnel, 8,400 of them sat down in the array as single GFP positive cells. That's 17% of the population. Of those 8,400 cells, 3,800 of them continued to expand and form GFP positive clones. That's 7.6%. And what was most fascinating is that of that 3,800 cells, 1,500 of them were actually RFP positive, meaning that over 2/3 of them, almost half, were not even antibody secreting to any real degree despite having GFP. So in total, roughly 3% of all of those GFP positive clones were RFP positive. What that means is in this consumable, 40% of the events that we observed sitting down as single cells were in fact clonal. That high degree of single cell to colony formation, again, is due to the productive environment where that shared media is supporting all of the cells. 41% of the clones were antibody secreting, and there were over 1,500 possible clones that you could use to interrogate to find the best hay in your haystack to use in your downstream production pipeline. And really what this turned into in the lab was from the infection of these cells all the way to clones was accomplished in a little over a week. And lastly, to go back to our comparison slide, in a single array looking for these high-producing antibodies, we were isolating 150 clones into 2 plates to give us enough cells to actually perform our downstream analysis. In that stack of 10 plates, we would have expected around 30 clones based off of those numbers. Now, this difference, 30 clones might be enough. For some workflows, that's probably plenty. But again, I want to emphasize the experimental burden, the user burden, the labor costs of handling 30 plates, 20 plates, 10 plates compared to 1 plate or 2 plates in the lab. So hopefully by now I have convinced you of the advantages for our forward screening antibody discovery approach using our instrumentation. In this workflow, we can perform this forward screening to identify and isolate only the antibody secreting cells that you're interested in. Rather than characterizing the entire haystack, we only have to characterize the needles. As I hope you've gathered from the last few slides, it's a very flexible workflow. You can use any antibody secreting cell that you wish, paired with any antigen and any capture reagents to create your tailored assay. It is completely scalable. Although in some of these examples, like the hybridoma and the 293T workflow, we used only 1 array because we had enough cells. You can upscale the number of consumables like we did with the B cell workflow to 4 or 5 or 6 or 10 or as many as you wish. And as I've emphasized several times already, it's incredibly cost efficient. It is going to save time, money, resources, and most importantly in this environment, effort. And if we want to fill the funnel, saving time and money and resources and effort is going to be very critical for getting us to that target antibody that we're actually interested in. So I'm sure by now you're curious how the technology actually works. So I'm going to spend the last few minutes giving you a bit more information on the technology and the instrumentation. As you've probably already gathered, the consumable works and acts just like a regular tissue culture dish. We can seed the cells into the reservoir of the cell raft array. The cells that we add into the array will settle by gravity into each of these microwell positions by a statistical distribution and attach to a growth surface within that microwell called a cell raft, represented by that light gold area here. In the single cell workflow, you could stop right here and isolate that single raft and go right to sequencing. Or in the clonal workflow, like the hybridoma and the 293s, you can continue to grow those cells in situ until they're ready for expansion and isolation. Using our software and all of the images that I've shown you in the talk today, we can very easily find the needle in the haystack. We can use the software to identify single cells at time point 0. We can perform a full image analysis of colony growth over time to ensure that the cells are growing at a rate or a speed that we're interested in. We can add in these fluorescent reporter antibodies to characterize the antigen-specific or non-antigen-specific production on a well-by-well and a raft-by-raft basis. And then ultimately, we can create a tailored triage list to map those cell rafts for recovery and downstream isolation. Now, I've said isolation throughout this entire talk, and I'm going to finally tell you how we actually do it. That's using our instrument, the cell raft air system shown here on the left. It's an incredibly user-friendly instrument, requires no fancy tools, no fancy operator. The scanning is incredibly rapid. We can scan in 4 colors, bright field, red, green, and blue. The scans typically take between 4 and 10 minutes, depending on the complexity of your assay. And when I talk about isolation, I don't mean sorting or dispensing or pipetting. What we're going to do is actually perform completely fluidics-free isolation of the cell raft itself. Not the B cells, not the colonies, the cell raft that the cells are attached to. We can do this either for the clones for expansion, or we can do this for single cells for downstream seq. So what this actually looks like in cartoon is that when we're ready to recover that raft, it is fully releasable from the array. It gets dislodged by the instrument, picked up by a collection wand, and then transferred over to a 96-well collection plate for downstream analysis. The raft will passively fall to the bottom of the microwell. If you're doing isolation for sequencing, you would stop here and go to processing, or you can continue to allow the cells to grow off of the raft, go back into suspension, and be processed for expansion, cryobanking, ELISAs, etc. So what this translates to in your lab or in ours, if you want to work with us to build out these assays, is a very accelerated, rapid forward antibody screen. Prepping the array and seeding the antibody-secreting cells is very fast. We can segregate thousands of cells in minutes. Seeding the array is no longer than basically 15 minutes of pipetting time. Once you perform your image acquisition, it takes 10 minutes over the course of 1 to 2 days. We use the software to identify those antibody-secreting cells, map the rafts for isolation, and isolating an entire plate takes roughly 45 minutes to an hour, depending on how many cells you're going to isolate. Everything we do goes into a 96-well plate, meaning that all of those plates are still compatible with any downstream assay that you're going to run for cell sequencing, ELISAs, plate-based assays, and can fit very nicely into any workflow that you're already running in your lab or any instrumentation. So with that, I'm going to close. I would be remiss to not thank our amazing collaborators and our team here at Cell Microsystems. Anna Lane is the remarkable research associate who has driven much of this work in our lab, and this project would not have been possible without her. We are eternally grateful to Jen Walker at Tetracord for giving us a real-world opportunity to test our budding assay in a setting that actually had commercial relevance. And of course, our collaborator at Duke, Dr. Zachary Hartman, who is our co-PI on the NIH-funded grant that funded some of this work, who was instrumental in all of the B-cell workflows, as well as his grad student, John Wang. If you're interested in learning more, I invite you to visit our website and to request a demo if you'd like to talk about getting this technology into your own lab. And with the launch of this workflow, we are also offering lab services where we can create custom cell line development for you using our instrument. We can help you accelerate your data generation, maximize your productivity, and then ultimately, when you decide to bring this instrument into your own lab, we can seamlessly transition our protocols to you. So with that, I'm going to close. I thank you so much for your time, and I hope you found this information interesting and informative, and I'm happy to take any questions that you might have.
Thank you, Dr. Hartman. That was some really exciting data. Really quick, before we get started with the Q&A session, we just have a quick poll question that we want to launch for the audience. And now that everybody's been clicking through, we'll move on to the Q&A session. So we've already received some questions that are very interesting from our attendees. The first is regarding the sequencing. Which method are we using for sequencing, and what's the percentage of B cells that you can successfully get sequenced from?
Yeah, that's a great question. We worked with a third-party vendor who made those recommendations to us. In this instance, we used specific heavy and light chain PCR and Sanger sequencing to just amplify the B cell specific sequences rather than something more global like a next-gen seq, and that worked out really well for us. Largely, the process is dependent on how good your nucleic acid prep is. I think in that particular experiment, we had about a 50% recovery rate of sequence, but we have since improved those methods by doing a lot of the prep upstream before sending the sequencing out. So I think what we've demonstrated in the past with the air system for single cell sequencing is that we have a much higher recovery rate of nucleic acid from single cells, because the cells are very viable and the system is very gentle. So I would expect a very high percentage of the B cells could be successfully recovered for downstream sequencing and informatics.
Great, thanks. So another question here. Could you use these workflows to establish monoclonal target-specific hybridoma lines after hybridoma fusion?
Absolutely. Even though we highlighted here the recovery of an already established line, there's absolutely no reason you couldn't do the same thing for a recently fused line as a modality to screen for antibody secretion. In a lot of ways, it's just as simple a workflow because you're still looking for a secretion of a single target IgG without needing to have that target specificity. And you could do that survival and screening right there on the array. So absolutely, you could start that process immediately after fusion and recovery at kind of any point in the process.
Great. And what about multiplexing? Can the assay be multiplexed to find multiple targets at one time?
Yeah, for sure. That's a great question. That is largely dependent on the antigens that you have available to you and the reagents that you want to multiplex. But hopefully, as was clear throughout the talk, the reagents themselves, they're all off the shelf coming from commercial vendors. And so it's the combination of them that makes the system work as opposed to anything proprietary that we're doing. So as long as you had a fluorescent detection reagent for whatever combination you're looking for, it would absolutely be possible. You can also coat the array with your protein of interest. So if you wanted to do sort of an actual kind of direct ELISA, you can also coat the rafts with protein and use that as a detection modality as well. But absolutely, multiplexing would be feasible.
And then this question here is about sort of the capacity. Is there a limit to how many cells that can be screened or isolated in one experimental run?
No, not really. The nice thing about the air system is that unlike other live imaging systems or live microscopes, these cell raft arrays do not have to be maintained on the deck of the instrument. So once they have been scanned in that 10-minute scan, they go right back into a traditional tissue culture incubator, which makes the workflow very modular, meaning you could seed one array or 10 arrays and still be able to process them all in a single day from a scanning and an isolation perspective. So you're really kind of only limited to as many cells as you want. And on a single array, the scale of the number of cells that you put on an array is largely dependent on that single cell resolution and your cell type of choice. So you could seed as little as 20 or 30,000 cells or as many as 50 or 80,000 cells, depending on your single cell resolution needs. Within a single day, you could certainly isolate, dozens of plates if you wanted to, because that isolation process only takes about 45 minutes for a whole plate.
Okay, and then one more question here. Do you use FP or FRET for the direct detection of the antigen antibody complexes?
We have not used FRET. We are using basically fluorescently conjugated primaries and secondaries to do so, as well as biotinylated proteins. FRET's an interesting idea. We have not tried that, but you could certainly give that a go. You can co-culture on the air system like you could in a traditional methodology. So if you had the FRET reagents and that was your detection modality, you could certainly seed at a higher concentration of the cells to ensure that you get multiple of the cell types on the array, and then look for that fluorescent recombination of FRET. But we have not actually tried it, but it's a very interesting idea.
Wonderful. Well, unfortunately, that's all the time we have for our Q&A today. As we conclude today's webinar, I'd like to extend our thanks to Dr. Hartman for sharing her experience, and to our audience, of course, for your active participation. We'd appreciate it if you could respond to a brief 3-question survey after the webinar has ended. A recording of this session, along with additional resources, will be sent via email and available on our website in a couple of days. And right before we sign off, please keep an eye out for our upcoming webinars on related topics. And thanks again for joining us today. We hope to see you for future events and hope you have a wonderful rest of your day.